Today we're going to be using these two cupcakes to represent spiked and not spiked skin cells on thorny dragons, with this being our hypothetical developmental network. So this is our wild type sequence, as you can see, where S promotes P, which then inhibits I, which then is not able to inhibit K, so K can produce E, which means that a dragon with spiked skin cells is produced. So now we're going to look at a few mutations starting with P. We're going to pretend that it's either mutated or not present at all. If this is the case, then P will not be there to repress I, meaning I will repress K, meaning that E will not be produced, meaning that no spikes will be produced on the skin cell, so we have a not spiked thorny dragon. We're going to look at a mutation in I. So the first part of the pathway proceeds as normal, but once I is reached, or we see that it's either not present or mutated, meaning that K cannot be repressed, meaning that E will be promoted, and that spikes will be promoted and produced in the skin cells of the thorny dragons. So next we're going to look at the strand being mutated in two different places, I and E. So if both I and E are mutated, S will still promote P, which then has nothing to repress, meaning that K will not be repressed, but also is not able to promote E, meaning that no spikes are produced on the dragon skin cells. Lastly, we're going to look at another double mutation in both S and K. So if S is not present or is mutated, P will not be promoted, which also means that it's not able to repress I. I in turn has nothing to repress, and there is nothing to promote E, which in turn cannot promote spikes on the dragon skin cells, meaning that the thorny dragon will not be spiky with this mutation. So moving on to the next part of the prompt, I'm going to be addressing the second bullet point and talking about how with a certain network, sometimes you can code for the same sequence, but some dragons might have spikes and some might not. So this can be attributed to something called epigenetic effects. So epigenetic effects mean that the phenotype is not caused by a change in a genotype, but instead it is caused by epigenetic changes which can have to do with the environment, cellular or outside of the organism, and it can add tags to different parts of the sequence strand and proteins, which can affect whether or not they are silenced or expressed. So, using this example, I'm going to be talking about an epigenetic effect called methylation. So methylation means that it adds different methyl groups to different proteins, and in turn, genes and sequences that are typically silenced are then expressed. So we can see this or attribute it potentially to this example, as this example does repress the production of spiked skin cells, but according to some epigenetic effects and methylation, it's not unheard of that these can't be removed and that the sequence itself cannot then produce spiked skin cells based on epigenetic effects and not a change in genotype.